Hello everybody, Sunday again. Okay, today continue our journey on biology. Okay, ready? Okay, we talk about A3.1 diversity of organism. Now we move on to unity and diversity in organism. Okay, not about the cell anymore, about organism. First of all, a concept is very clear. Variety of organism. Look at you and your friends. Okay, all the human beings. Okay, no individuals are identical in all their traits. Okay, we have some difference. Even the twins, and when they develop out, they have some different traits, right? So the pattern of variation are very complex. Okay, you and your friend are human being. We are the same species, but we are a little bit different. Okay, and the differences, the variation, are complex. Okay, and then first of all, we have a different concept of species. First of all, is the morphological concept. This concept is said that a species is a group of organisms with a shared trait. And Linus originally used this morphological concept of the species about shared traits. Okay, shared traits is the first morphological concept of species. Now the species concept will change later. First one is a morphological traits, and we are from this morphological system. Okay, we developed the binomial naming system. Okay, and the naming, all the names have two names. Okay, and the name from the Latin words. Okay, those are scientists like the Latin spring. Okay. So each name has two parts. So the first part is a genus, and the second part is a species. The genus name given an initial capital letter, okay, and the species name is a lowercase, so you can distinguish them easily. Uppercase, genus, lowercase, species, okay. And the species in the same genus have similar traits. Okay, so the same first name, okay. Genus means closely related. For example, we are Homo sapien, and all our friends are closely related has a genus Homo. Okay, but we are closely related, but we are different species. Okay, and different first thing may mean that they are different. They are different genus. Okay, they are different. Okay, for example, human and monkey are different because we have different genus. Human is Homo, and for the monkey is a Semiforme. We are classifying into different genus. Okay. So when new species are discovered, first of all, they will find out their first name, okay, the genus first, okay. Classify that genus, and then they will recognize their species according to the International Code of Zoological Nomenclature. There's some rules on it, okay. And then we have another biological species concept. It's not about the traits, okay. It's about species is a group of similar individuals that can interbreed in nature and produce a further offspring. They need to interbreed and produce further offspring. So from this definition, you got it. Some are not solved exception problem. Hybrids, okay. Microorganism, plants, okay. They don't do sexual reproduction, okay. They don't interbreed, okay. And bacteria and organism, okay. They reproduce asexually. They are not included in this concept of species too. So every concept of species has its strength and weaknesses. The biological one has this one. For those that don't interbreed, they are not species according to this biological concept. Population and species, another concept. Okay, so we have species, and species can separate in the world in different geological situation, site, okay, place, and we call it a population. Okay. Population is a group of organisms of the same species that interbreed and live in the same place at the same time. Okay. In the time and space they join together, they live together, we call it a species. We call it a population. Okay. Population is smaller than the species. Okay. It's difficult to distinguish between populations, okay, and species, okay, due to divergence of a non-interbreeding population during a speciation. And what is speciation? Speciation is the splitting of one species into more, two or more, okay. So the concept is like this. So we have, for example, first as one species, okay. They live in different populations, and they are separated. So later on, they develop their special traits, and later on, we found out that they are quite different, and we name them as a different species. Okay, so population of the same species become more and more different in their traits, and then eventually we'll call them these two population, two species. We'll discuss this speciation later. And variation of chromosome number. For example, in the microscopic point of view, in the DNA point of view, we can distinguish species in this way. Okay, for example, in a different number of chromosomes. Okay, but there's some difficulties. Okay, for example, human has 46 chromosomes, 23 pairs. Okay, chimpanzee have 48, but potato also have 48. So if we just distinguish by the number of chromosomes, it's troublesome. Okay. Our animal chromosome can range from 254 to 2, okay, in some worm, okay. And the variation of chromosome number is higher in plants. Okay? For example, some fern has 1,260 chromosomes. So don't think that we have a more number of chromosomes, so we are smarter. No, 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 no. Don't think that this fern is super smart. No, no, no. So the diploid cell, okay. Remember, we need to have an even number of chromosomes for to have a fertile offspring. Karyotyping and karyogram. This is some way to tackle or numbering the chromosome. The chromosome have different characteristics. Okay, different chromosome has their individual characteristics. For example, the banding pattern, they have a bright and dark bands. Okay, different length and different position of the centromere. Remember, a chromosome is a pair and they are joined together at the centromere. Okay, but the position of the centromere is not necessarily at the center, at the middle of the chromosome. Okay, so they have a different banding pattern, bright and dark transverse band. They have different names. Okay, of course, with a few chromosomes, they need to have a longer chromosome, right? Fewer chromosome, longer chromosome. Very funny. Okay, and the chromosomes are joined at the centromere, but not necessarily at the center. If it is really at the center, we call it a metacentric chromosome, really near the center, joined together by the centromere. If it is not, we just say that it is a sub-metacentric chromosome, not at the center. It's off center. Off center is sub. Metacentric is normal. I shouldn't say normal. It's the one at the center. And we have a classification of the chromosome. Okay, chromosomes are classified according to the banding pattern, length, and centromere position. So we have three criteria to separate them. Okay, and karyotyping is a process for the pairing and ordering of all the chromosomes within an organism. Okay, by karyotyping, a karyogram is drawn. Okay, this karyogram is drawn according drawn the chromosome as pairs, and then draw them from a long to short length. Okay, so the longest one is number 1. Okay, so shortest one is the last one. Okay, so different species have different karyograms. Okay, so karyogram can be used to distinguish a species quite effectively. Okay, karyogram can be used to figure out the genetic abnormality too. Certainly, it has 3 genes. Okay, there. Okay, 3 chromosomes. Oh, then you find out it's a problem. Okay. And the human, for example, human chromosome number 2 is very funny. Okay, its chromosome 2 is joined by a head-to-head fusion of 2 old chromosomes, and these 2 old chromosomes is still separate in other species. Okay, so it's very funny, but we don't know why they are fused. Okay, what happened? Okay. And then unity and diversity of genomes within the species. Like a genome is a concept. Okay, it means that all the information, all the genetic information of an organism, we call it a genome. Okay, and within the species, the unity of genome is quite rare. Okay, organisms in the same species share most of their genomes, most of them, but they can have the single nucleotide differences, mutation. We call it a SNP. Okay, SNP is a kind of mutation. So there is the diversity within genome can be as small as just 1 nucleotide. And the gene pool, okay, is all the genes in an interbreeding correlation at a certain time. So genome is big. Okay, gene pool is all the genome joined together at a particular time and population. Okay, all the genome joined together, we call it a gene pool. Okay, it contains all the variation of genes in that population. Okay, so different we can say different people at different time have different gene pool. Okay, the old time and the modern time, we can have different gene pool. It's a little bit different. Okay, eukaryote genomes. Okay, variation of eukaryote genomes. Okay, most bacteria genomes are circular. Okay, but for us, eukaryote genomes are linear. A genome may vary in overall size. Okay, which is determined by the total amount of DNA. And genome also can have a vary in the base sequence, of course. Okay, are not so different. out. Okay, and genome variation between species can be even bigger. Okay, so within the species, genomes are very similar. UMB genomes are very similar, but between us and other animals, other species, the genomes are completely different. Okay, can be. And then we can study, okay, we call it comparative genome, study the genome of different species. Okay, if they are very similar, we are closely related. Okay, if the genomes between two species are quite different, so they are maybe they are not genetically linked. Okay, so if two organisms have a close common interest, okay, their species genome should be more similar. Right? So by comparing a different species of genome, we can know how they are related in genetical relationship. Okay? We call it comparative genomic. And the size of genome, okay, we have units for it. Wow, funny. Okay, we call it GBP. Okay, gigabytes base pair. How many base pair are there? 10 to the power 9 base pair. We call it 1. Okay, and genome size varies within species. Okay, for example, in female, a little bit more than male. Okay, 6.37, and for the male is 6.27. Just a 3% difference. Okay. Genome size varies much more between species, of course. For the plants, it can be as small as 0.06, and for the another Japanese plant, it can be about 152. Okay, wow, so quite a big difference. So don't think that the more genome, the more chromosome, more number of chromosomes is marked out. Okay, no, there's no relationship. Okay, complexity, okay, and the genome size, number of chromosome are not related. Okay. Okay, actually, on the contrary, in some research, we found out the plants with giant genome size. Okay, it is a little bit not so adaptive to the polluted soil or the extreme environment. So we expect that this kind of a giant genome size species will be extended quickly. So we call it rare. 152, where we can't find it. If we can find it, where in the natural environment for these Japanese plants. Okay. And the whole genome sequencing, we want to find out the genome of human beings. Okay, they have a lots of steps that we have to do. Okay, because genome is quite big. Okay, first of all, we have to isolate the DNA, right? And then we want to study them fragment by fragment. So we have to randomly fragment them. Okay, make them to be a smaller fragments. Okay, then we can size selection. Okay, sort out their size. Okay, and then we have the library to store these kind of fragments. And then the last, we have to join these fragments back together by the paired and the sequencing method. And they went through this breaking, study, and joining back procedure, and the genome is assembly. And then thanks to this formalism of this methodology, this part, each step can be done at different approach. Okay, and it is an international collaboration efforts. Okay, for the genome sequencing. It launched as the 1990 October. Do you know how long does it take? It takes about 13 years, 12 and a half years for it to be completed. Okay. So remember, 13 years is a huge time. Okay, but the technology doesn't grow yet. Okay, and the whole world work together, all the laboratory work together for 13 years. But the technology evolved quickly. Nowadays, whole genome sequencing is quicker, easier. Okay, and then we think that potentially we can use this application. We can know different people's genomes, and we can personalize the medicine for everyone. Okay, and then it's increasing speed and then decreasing cost. Okay, and remember before it's about 13 years, now it's 5 hours. I think it's quicker now in 2024 due to the AI. Okay, by the advancement thanks to the AI. And now it dropped from $1 million in 2007 when it first commercialized, and now about 2023 about $500. Okay, I think it will drop it even lower. You can have your own genome one day too. Okay? And then next part is AHL. Okay, first of all, is about the biological concept of species. We have to think about it in AHL. Okay, for example, for the asexually reproducing species, it doesn't apply to it absolutely, right? For example, the worms and the sea stars. Okay, and the bacteria. Okay, so this is the reflection on the biological concept of species, so they require successful interbreeding to fertile offspring. It doesn't work for some of them, okay? And this some of them is quite unlogical, so we don't call them exception. And also, this doesn't work for bacteria. So we call it a horizontal gene transfer. For bacteria, they are very adapted to the surrounding because they can share the DNA with each other. With different species of bacteria, they can share the DNA quickly. Okay? Don't let the recipient can be different species of bacteria. So it's hard to distinguish the a gene pool or genome of a bacteria is very quite unstable. Okay, so it's hard to apply the concept of biological species concept to them, and even for the genetic one too. And the share of a species. Okay, this is the first concept, right? But it is hard to sometimes. Chromosome number is a share of within a species. Okay, and crossbreeding between closely related species is unlikely to produce fertile offspring. Right? If parent chromosome number are different. Okay? Although they are closely related, but when they interbreed, the offspring is not fertile. For example, a mute is a cross-breeding of a female horse and a male donkey. They are quite similar, but a horse has 64 chromosomes, donkey 62, and they mix together, add and divided by 2, they become 63. Not an even number, you know. And this is the problem. If it's not an even number, they can't be divided into pairs, okay, so they can't reproduce. So male is infertile. Even the close one, sometimes they can't interbreed if they are different species, okay. And we have another way to distinguish between species. This is something that we needed to do it in the laboratory or during the lesson time. Okay, we call it dichotomous key. So dichotomous key is tools as a binary system as the tools to identify specimen, and we use this tool apply to different species. Okay, in each step, okay, they are divided into two parts. Okay, yes or no. Always yes or no question. Remember, yes or no. You can't be a other thing or not. Can be red or yellow. No. Red or not red. Okay, always yes or no. Okay. Question. Okay, can using this step. Okay, step 1, step 2, step 3, and step 4, and then they can distinguish a lot of species into different species. Okay. In IBDP, we need to try to play this game. Okay, and apply this knowledge, this methodology to local plants or animal species to develop a dichotomous key. It will appear in the exam question too. So we need to have some experience on it. Remember during next time. And then it is also useful to use this DNA way to investigate about diversity in habitat. In habitat, there are different species living there. Okay, but sometimes we can't see the species or the old time species. Okay, so we need to find out which species live there or not. Okay, in the habitat by the fragments of DNA living there. Okay, they live there. Maybe they have some fragments that live there too for the DNA, right? So from the environmental DNA, we just find out the fragments of the species. Okay, so we need a technique to find out the species from this little fragment. We call it a DNA barcoding. DNA barcoding is a skill, okay, that we can just find some DNA extractions, just find a little bit, okay, it's just a small fragment of the genomes. And from this DNA barcoding skill, we use a PCR amplification, duplicate it a million times or 10 million times. So there's fragments can study that sequence quickly or a lot of times, and then we can find out which species are there. It is really useful, okay, especially when the species only leave fragments, small fragments of its DNA in the environment. But by this we can study who live in the habitat before or now, okay, for a long time or for a short time, okay. And for a short time, we know that it should be the invasive species, okay. After all this knowledge about the species, the existing, the old one, invasive one, okay, we can have a more design or more planning to conserve the biodiversity there, okay. Although maybe we can't see the species so frequently, so we can get the species who are living in that habitat by this kind of fragment of DNA through the DNA barcoding skill, okay, extraction, amplification, and sequencing skill. So we can study habitat, okay. This is an application of DNA. Okay? DNA is a microscopic thing. By this microscopic skill, we can study the macroscopic thing. And this is the magic of the science. Okay? Thank you. Thank you for going through this part with me. Okay. If you like our channel, remember subscribe and share with your friends. Okay. We want more people to hear our voice. Okay. And eventually maybe we can help them too. Okay. Thank you. See you.